Okay. All right. So for our next presenter, uh, Dr. Sharif is in an airport right now, so he had some flight delays and so was unable to make it. We have one of his graduate students, Somye. Oh,
Somaye Mohammadi.
right. Well, I'm going to have you come on up and let you start your presentation. And we have that. Oh, yeah, she can use that mic. Is that one? Yeah.
Good morning, everyone. The title of the presentation is PFAS contamination in water samples and detection technologies. First, I'm going to talk about a background about the technology and PFAS importance detection. You know, this slide shows the ongoing global news about PFAS contamination in water samples. And the recent news articles report this PFAS contamination that shows the importance of monitoring of this PFAS contaminants in water samples. As you can see in one of the news, a study done in Japan, PFAS found in 20% of tap water. And, it community impact shows the high level of PFAS in low income, areas. And there is a link between exposed to this kind of contamination and different kinds of disease, not only for adults, but for unborn babies too. Uh, PFAS, it stands for poly and perfluoroalkyl substances. As you can see in this slide, it belongs to a large group, more than 6,000 man-made chemicals, and around 6 million of US citizens exposed to this contaminants by drinking, contaminant water. And you can see some common structure of PFAS compounds, and you can see all of them has CF bond, and it's the kind of a strong bond that makes them persistent, and they can accumulate in environment and enter our food chain and accumulate in wildlife and human body, and they are dangerous and toxic. And here you can see the diverse application of this chemical. Because of heat resistance of these chemicals, they are used in non-stick cookware, and they do not readily biodegradable. And they can accumulate in organism, living organism like polar bears. And if you saw in another previous slide, they have carbon, hydrogen, nitrogen, and oxygen that creates a polar end and make them, chemical stability. So it's really important to find a method for, remediation, monitoring, and detection, low concentration and amount of this chemicals in different, environment, especially in water systems. As you can see in this slide, in the left image, you can see the military, personnel using aqueous film forming foam for fire suppression. As military sites is a major resource of, PFAS contamination because of using this kind of AFF compounds. And PFAS do not, evaporate easily, and they do not break down easily, and they can, accumulate in environment. And in this picture, you can see the absorption of this PFAS on the soil particle, and the plant can uptake this kind of compounds via roots and bioaccumulate in this kind of plants and enter our food chain, enter our body and accumulate in our body. And because of capillary action, this kind of chemicals can move deeper into water system, groundwater. And in a US map, you can see, different color, site, especially in eastern part of US. The water sources show in light blue color. It's above, PFAS proposed limit. And the dark one is below that proposed PFAS limit. And the purple color shows the military sites, and the orange shows the other sites. So it's really important to, the this design a method, a technique for detect this kind of chemicals or remove them from our environment. And here you can see the complexity of PFAS in terms of chemical, physical, and human exposure pathway. And for chemical forms, you can see this kind of chemicals can be anionic, cationic, zwitter ionic, and neutral. And for physical, structure, they can be straight, branched, and oxygen bridge bond and cyclic. And in these two image in right side, you can see the level of blood PFAS in manufacturing workers or the people who work in contaminated sites is much more higher than general US population. And PFOA and PFOS have declined using them since 2002, but still their contamination remained. And here you can see the PFAS toxicity versus mobility. And there are two terms, one reference dose in y-axis and, the coefficient of partition soil water for mobility. As you can see, the red color shows the high risk and the green color shows the low risk. And for a low amount of R, RFD and KOC shows the highest risk and high toxicity and high mobility. And the yellow side for you can see for PFOA and PFOS, it shows the medium toxicity and mobility. But the green side shows the low risks. And here you can see the health issue through drinking contaminated water. As you can see, there are two different kinds of line, the solid line and the dashed line. The solid line is for high certainty. It means there are enough evidence. And the dashed one show the low certainty. It means requiring more evidence. And it affects adults and unborn babies. For unborn babies, like lower birth weight and decrease vaccine response and increase miscarriage risk. And there are different kinds of disease risk, like cancer, like organ damage, thyroid disease, or high cholesterol and obesity. So it means, exposed to this contamination is really highly risk for human. And in this slide, you can see health and environmental concern in US states. As you can see in box in the left side, the health advisory limit was issued in 2016 for 70 ppt, P4A and PFOS, but it was revised in 2024 for very low concentration, around 4 ppq for P4A and around 20 ppq for PFOS. And this bottle shows the, sample, PFAS samples source like air, like water, like soil, food, and, the exposure pathway. As you can see, the map of United States in three different color that shows PFAS sampling. For white color, it shows not actively used PFAS sampling, but the blue one used the EPA guidelines for PFAS sampling, but the red one has a intense regulation, use both EPA and state guideline for PFAS sampling. And here in this slide, you can see a research for investigating PFAS sources and variation in Rio Grande, and they detect different PFAS, as you can see in box, the diagram in different color in water samples. And, if Fourier transform, ion cyclotron, resonance mass spectrometry is a very high resolution and accurate technique for identifying similar PFAS compounds. And as you can see in left side image, there is the world largest mass spectrometry, 21 Tesla in, in Florida, in National Magnetic Field Laboratory in Florida. And this technique can differentiate some, PFAS compounds with similar, uh, uh, mass. And as you can see, it can be used for detecting PFAS in, complex matrix like AFFF. And you can see the mass spectrum here, that this technique can resolve this small mass difference of PFAS compounds. And here you can see the site sampling and sample processing in three different sites. One is groundwater sample from three US, Department of Defense, and another one wastewater treatment plants samples, and the third one AFFF samples. And there are control for these samples because to be sure the sample are not contaminated during collecting. And liquid extraction was used for extract this PFAS from sample, and the solvent was evaporized to concentrate the PFAS and then reconstitute in high purity methanol for further, mass analysis. And here you can see identification of dissolved natural organic matter in three different sites. As you can see, this peaks for, wastewater treatment plant is the highest one, and for groundwater is high too. But the lower peaks is for the aqueous film forming foam. And here you can see how confident we are in high resolution mass identification. based on the, the data requirement, we have five levels of confidence. For the first level, confirmed structure, we need a mass, tandem mass, retention time, and reference standard. But for the last one, that exact mass of interest, the just minimum data required is the mass. And here you can see this Fourier transform and cyclotron resonance mass spectrometry, could detect PFAS in 11 samples. But as you can see, the highlight blue, it shows around 300 novel PFAS with high confidence. And they classified as a 75 classes, and around novel PFAS, compounds around more than 1,700, and other novel potential PFAS is more than 6,000 compounds. And as you can see, these three different methods help scientists to determine where these PFAS compounds come from and how they behave in the environment over time. The first graph, you can see it shows their behavior over time. They will be oxidized by, or reduction or retardation. And the second graph is for principal component analysis, a chemometric method that shows the PFAS compounds can groups together. And the third one shows which kind of compounds in three different sites are unique and which one are shared in 2 or 3 different sites. As you can see, for example, for AFF, around 192 PFAS compounds are unique. But, for around 25 are shared in 3 sites, but from 6.2 till 17% are shared in 2 sites. And now I'm going to talk about the research I did in Dr. Sharifan labs. And the first research, we designed a fluorescence assay by synthesizing fluorinated carbon dioxide for PFOA detection in groundwater samples. And we published it in ACS Applied Nanomaterial in September 24. And I did 3 different research for PFOA detection. The first one by synthesizing fluorinated carbon dioxide. The second one by, a modification sodium alginate hydrogel with fluorinated carbon dioxide. And the third one, I did this research by help of aid of one of our undergrad students. And we modify ferromagnetic polymer with nitrogen dope carbon dioxide. And here you can see the steps of designing the sensor for PFAS detection. The first step is synthesizing the nanomaterial or composite. Then we need to characterize this composite or nanomaterial by a spectroscopy technique, electron microscopy, or zeta potential to figure out which kind of functional group we have, which kind of bound we have, and how we can modify to improve sensitivity and selectivity. And then testing other PFAS compounds, and then plotting calibration curve, and finding the lowest concentration we can detect. And the last part and the most important part is field application. I mean, detecting of this kind of target in real sample, like groundwater, like well water or tap water. Why we choose carbon dioxide? Over 10 years, I have experience in synthesizing, fluorescence nanomaterial for biomedical and environmental application. And carbon does gain a lot of attention because of their synthesized procedure is not complicated, and there are different kind of precursors for synthesizing them. And if we use the green precursors, we have biocompatible carbon dioxide, and they have functional groups, and we can modify them with other groups too, and they are photostable. And Then, by choosing good precursors, they have potential for different kinds of interaction. We can, for example, choose this kind of precursors we can use to have electrostatic interaction, hydrophobic interaction, and we choose polyethylene amine, a polymer for providing electrostatic interaction, and tetrafluoroterphtalic acid for hydrophobic interaction. You can see here two different synthesizing methods. The first one for fluorinated carbon dot and the second one for non-fluorinated carbon dot. And we use the solvothermal method. It means we use the solvent, it's not in aqueous solution. It's a kind of, we use organic solvent here. We use ethanol for synthesizing. And by changing the Mohr ratio of tetrafluoroterphtalic acid and polyethylene amine, we synthesize different kinds of fluorinated and non-fluorinated carbon dot. And under UV image, this carbon dot shows the blue color. And for proving this electrostatic interaction, we measure the zeta potential to see the surface charge of carbon dots, and it was 17 millivolt. And after adding PFOA, as we expected, the zeta potential decreased to around 6 millivolt. And here you can see the FTIR, spectra, and we can see different kinds of bond like CF, C double bond oxygen, C double bond C, C bond nitrogen, nitrogen hydrogen, and nitrogen oxygen hydrogen that improve the solubility in water. And sorry. And the last image is the UV spectra of the carbon dots, and we have the two bonds, one for pi to pi star transition for carbon double bond carbon, and the second one around 130 nanometer for nitrogen or non-bonding to pi star transition. And it's really important for fluorescence technique to find the optimal excitation wavelength. And we scan this fluorinated carbon dot from 300 nanometer to 400 nanometer. And the optimum excitation wavelength was 340. And you can see the TM and the size distribution. And as you can see, the size of the carbon dots was lower than 7 nanometer. And for elemental composition.
Yeah, yeah, we do. Yeah.
Okay.
And can you look for your last slide?
Sure. Yeah. And we detect the PFOA in two water samples, the tap water and groundwater. And, we spiked 3 different concentration of PFOA, and the recovery was, acceptable from 96 to 102%. And I will be happy if you have any question, I will take.
Yes, thank you.
Thank you so much.
We are going to have to forego questions now. Maybe for those who want to stick or able to stick around, if you have questions, but we're going to get to Dr. Kong here for our final presentation for this morning. And I think we have. Thank you.
Yeah.